All right, so this is the fifth and last installment of your Evolution of Populations lecture. I erroneously stated on the previous one that it was the third. I apologize, that was the fourth. Okay, so this won't take too long. Well, there are limits to what selection can accomplish. It can only edit what exists. It's limited by historical constraints. And when there are adaptations, there are always compromises, and you're dealing with the interactions of chance, natural selection, and the environment. So let's take a look at what those limitations are. Genes can have multiple effects. Remember that. Alleles can affect many aspects of phenotype, and what we're talking about here is this idea of pleiotropy. We've talked about that before. This limits how much the phenotype can actually be changed. Selecting for example, large clutch sizes in chickens leads to eggs with thin shells. So now we can't really select for chickens that lay many more eggs than they do now, cows with more muscle mass, nor corn plants with ears at every leaf. Just not possible. And remember that evolution requires genetic variation. Only 31 ancestors contribute to over 80% of the entire gene pool of modern racing thoroughbred horses. That's kind of scary. And race times have not improved drastically over the past 50 years, despite very, very highly selective breeding. And what you're looking at here are the Kentucky Derby winning times in seconds, because it goes by so fast, over the last 100 years. And you can see that it really hasn't changed much here. Of course, going down is good. That means they're getting faster. So genetic variation has been removed by this very selective, intensive selective breeding, and now no significant change can occur because of the fact that when we go back, only 31 ancestors contribute to most of that gene pool. But not all not all phenotypic variation is genetically based. Here's an interesting example. Compound eyes in insects called ommatidia. So if you're looking at all of the little islets on one of their compound eyes, are actually called ommatidia. And we frequently see a difference in the number of ommatidia between the left and right eyes. Now, scientists cannot experimentally create consistent lines of flies with more ommatidia in either eye, because there's only one gene that controls the number of ommatidia for both eyes. So one gene for both eyes. And in fact, what they've discovered is that the differences actually arise from something that occurs in the developmental process. So it's not genetically based. And gene interactions can also affect the fitness of alleles. Remember epistasis? This is where an allele for one gene may react differently depending on the effects of the alleles at other genes. Alleles can impact the effects of alleles at different loci. Remember that genes can affect other genes? Now, a selective advantage of alleles might vary with different genotypes. They may not always be that advantageous. For example, Hartle from Harvard with his team studied bacteria with the gene 6-PGD, which is an enzyme that metabolizes gluconate. And polymorphism does exist at the 6-PGD locus, but no alleles have a selective advantage over the others. They all seem to have the same effect, and all the bacteria grow at the same rates. A second gene controls an alternative biochemical pathway for gluconate metabolism. And this uncommon allele disables the second pathway so that the 6-PGD enzyme can metabolize gluconate. So even though there's this other allele, it basically gets disabled or turned off. So bacteria with several 6-PGD alleles were superior to those with bacteria with just fewer of the alleles. So that's kind of interesting. So we're going to end this with this little statement here. The ability of selection to produce evolutionary change is hindered by a variety of factors. So in summary, all that we've covered is gene variation, the Hardy-Weinberg principle, agents of evolutionary change, measuring fitness, how evolutionary forces interact, the different types of selection, and limits to selection. And all of this was pretty dense, but it will serve you well when we do our population genetics lab.